Hello, everyone. My name is Malcolm Guzman, and today we're going to talk about tight junctions. Now, what are tight junctions? Tight junctions are these little stretches of protein that you can find in between the cells in the epithelial layer and other epithelial cells that prevent the leakage of molecules from one side to the other. From the apical region, which if you picture your intestine, the inside of your intestine is the apical region. They prevent any sort of molecules from entering from that side into your basal region or the region that comes into contact with the rest of your body. And how they form is like if you think about two sheets of corrugated metal with those little ridges, if you bring them together, they're going to touch at those ridges, and that's how tight junctions form, is at those little ridges. They are made up of occludins, IGSF junctional adhesion molecules, and the most important part that we're going to focus on is claudins. Now, claudin proteins have these 4 transmembrane domains, including this large loop right here. And in this large loop are charged amino acids that interact with the charged amino acids of the claudin loop in the adjacent cell. And this creates the seal that prevents molecules from passing through, and we get our seal in between the apical and basal regions. Now, this bond is not entirely impervious. There is some sort of these amino acids do form a sort of selective core that allows certain ions to pass, and that's how we get paracellular transport, which is the transport of certain ions in between the cells instead of having to go through the cells themselves. These tight junctions are primarily found in the intestinal and epithelial cells, like we talked about before, in the intestine, the glands and the ducts that have to come into contact with the digestive system, the bladder, where they prevent urine from leaking into the rest of your body, which is really important. And they're also responsible for the blood-brain barrier that we see in the endothelial cells in the brain blood vessels. I included this little figure right here where you can see an experiment in which scientists introduced an electron-opaque tracer into one side of a layer of epithelial cells, and you can see that the tracer was able to leak into the spaces in between the molecule and in between the cells, but stop right where it hits that tight junction, and that's what creates that seal that we talked about. Tight junctions also prevent the lateral movement of lipids and proteins within the cell membrane, so that certain transport proteins and stuff like that are able to maintain the required concentration in a certain area without drifting all over the place. Both of these aspects are what we call the barrier function of the tight junction. But the tight junction also serves what is called a fence function. And this fence function has to do with maintaining the apical and the basal region segregation and maintaining cell polarity. So the side that's supposed to face the apical region always faces the apical region, and the side that faces the basal region always stays in the basal region. Now, what is this important? It's important because tight junctions actually play a really big role in the development of cancer. Now, what happens in cancer is that cancer cells have a reduced integrity in their tight junction function. And so what happens is that when you lose tight junctions, what happens is that the cell polarity is compromised and you start having ligands that are supposed to stay in the apical region that promote cell growth entering the receptors in the basal side of the cell. And so you get this autocrine action where the cell is telling itself to grow uncontrollably, and that's a very bad thing. The other aspect of this loss of tight junctions is that the Hibba pathway regulators, which is this pathway that controls the growth of organs, and Merlin gene tumor suppressants that prevent the development of tumors, have nowhere to bind to if these tight junctions are not there. So this also adds to the uncontrolled cell growth, and then we get what is called epithelial mesenchymal transition. Now, mesenchymal cells are cells in early embryonic development that are sort of stem cells that allow cells to move and go where they're supposed to go. But when this happens to cancer cells, that's when we get metastasis and the cancer moving around and growing all over the place. So tight junctions are actually a really important area of study in analyzing how certain cancers that affect epithelial cells are treated. Now, another finding that I found in the study was how tight junctions interact with the damage caused by aspirin. Now, as you know, when you take a lot of aspirin for a extended period of time, it can actually cause lesions in your gastrointestinal system. So what the study did is that they took this drug called mozapride, which is usually used to treat gastritis, gastroesophageal reflux disease, functional dyspepsia, and irritable bowel syndrome, and they gave three groups of mice, control, a group that was given aspirin, and a group that was treated with mozapride, and all three of these groups were fed a lot of aspirin over 4 days. And what they found was that the group that took aspirin suffered from increased lesions, reduced tight junction integrity compared to the control, but then the mozapride group suffered fewer lesions. It actually had increased occludin expression, occludin, one of the proteins that plays a part in tight junctions, and tight junction integrity was unaffected by the consumption of aspirin. So what the study points to is the fact that mozapride can actually be used to prevent the damage caused by aspirin because it allows these tight junctions to maintain their integrity and for occludin to be expressed at normal levels, even when exposed to aspirin. So what I'm wondering here is if these claudin meshes are so good at keeping molecules in and out of places, could claudin-based meshes be used medically or surgically in cases of organ injury to prevent leakage of fluids inside the body? Or do we have synthetic methods that are less intrusive or more effective still? Is there anything there, is what I mean, we can use artificial claudin meshes in any other applications in medicine? Not included in their patient references here, but we also have references in each slide. Thank you very much for watching, and I hope you enjoyed. Thanks.